Okay, so we're going to look at the classic experiments of biology. In biology. And we're going to keep looking at these classic experiments because there's a bunch of them. In this case, we're looking not at the transforming principle, which was the first one. We did look at the Griffith experiment, right? So again, the transforming principle is kind of a checklist of what you need to know for the exam, right? Transforming principle, so you have to understand the experiment. Right, and part of that understanding of the experiment is you understanding what the result was. Understanding the steps of the experiment, including and especially control groups and experimental groups. Now, I just want to wrap up this experiment so we can go on to the next one. And one of the things I want to point out is that you start with an environment. And the environment in this case was around circa 1920s, right? 20s and 30s. And we're dealing with before antibiotics. And really the key to this, understanding what's going on here, the key to understanding what's going on here is that Griffith started with one question. His question was, he was studying, I don't know what his exact question was, but he was studying pneumonia, right? Help me. So they were studying pneumonia because there were no antibiotics, right? So before antibiotics, this bacteria was killing so many, right? So that's a really big idea. Why we do science? Why are we studying these things? And in the middle of studying pneumonia, what he found was something interesting, something that challenged his understanding of how things worked. There was something transformed the rough to smooth. He found a transformation, which led him to ask a different question, a new question, and that new question was, what was that? What transformed? The bacteria, which led him then to do what? Can anybody answer that? To experiment, that's right. It led him to design a new experiment. And that's science. You see, we like to tell you that science is you come up with a question, you do a hypothesis, you design an experiment, you test that hypothesis, you collect the data, you analyze the data, you get to a conclusion. But if you look here, in the middle of his experiment, he discovered something, a transformation, which led him to jump to a new question before he was finished really with the conclusion of the first question. It gave him a new question to start looking into and designing a new experiment. And that's the real world. So you're doing something and what was it? Someone was the discovery of the first antibiotic, penicillin. A scientist was working on, and I don't remember the details, but he was working on one question, and he left a sandwich on the desk. At least one of the stories is he left a sandwich on his lab bench. He came back the next day and there was mold on it, and he noticed that the mold, no bacteria grew around the mold, right? So that led him to think, wait a minute, let me stop doing why is that happening? So it's curiosity, right? What led, we call this a serendipitous discovery, right? Something that's very lucky. He didn't plan on discovering penicillin, but by looking at the impact of that mold, in fact, so you should know that penicillin was made by Penicillium, which is a fungus, a green fungus. And that fungus that grows on bread, by the way, creates an antibiotic to fight off bacteria. And that was the first discovery of an antibiotic. And since then, we've had a lot of adaptations. So penicillin was one of those first discoveries of the first antibiotics, the first class of antibiotics. So the point is that this is science. This is the real scientific method. Yes, we go through the steps. We try to go through all the steps so that we remind ourselves that we have to be objective and that we're looking at facts and we're comparing facts and we're analyzing facts. And we're making sure they are facts and not assumptions, right? Because unfortunately, as human beings, we love to make assumptions because assumptions work, right? I can assume that something, and if I assume it, most of the time it's right, I can go with that, because trying to analyze everything is just too much. So it works for humans to do that, but in science we have to not do that. So it's interesting that this is one of the big takeaways, one of the things that I will be testing, and one of the things you should know, that the whole process, the scientific process that I just described to you. But I do want you to know the steps of the experiment. I do want you to know the control groups, and I went through it on the video and I went through it in class, and I want you to know what the experimental groups are. I want you to know that this was a big discovery. It was not only a beautiful experiment, it was also a giant discovery, and the giant discovery is that DNA is the transforming principle. Let's say, instead of principle, let's say transforming molecule. And you do need to know, remember that, they thought that it was protein, right? Why did they think it was protein? Yeah, it's okay. It's a good answer. That's fine. It's because there are 21 letters, right? And if you're going to come up with the stuff of life, the things that explain who you are, as complicated as you are, and all the different species that are on the planet, you would think you'd need at least 21 letters. But actually all you need is 4. 4 little letters. A, T, G, and C. Those are the only letters you need to make DNA, to code for everything that is alive today on Earth. So very interesting. This was a shock. This is another part of the process of science. This discovery proved himself wrong in more than one way. He had assumed that remember this one step in the Griffith experiment, that he had assumed that mixing up of the dead strain of smooth bacteria with the rough bacteria injected in the mouse would lead to that mouse being alive, and ended up being dead. That was wrong. He was wrong in his initial prediction, and he got excited about that. He got excited about that because he saw an opportunity in his failure, right? So many of us are scared of failure, right? But in his failure, he failed big, and what he did is he got excited because that meant that he could look into the transformation, a bigger problem that he could maybe come up with a solution for. And in the end, he ended up proving himself and all his teachers and all his colleagues wrong. Wrong. Big failure. They had wasted years thinking that protein was the language of inheritance, the language of transforming things, right? When it actually was DNA. They were very excited though, very happy, because that failure allowed everything that you know today to occur. All the genetics that we know today is because of that giant failure that led to a new discovery. So you need to stop being afraid of failure. You need to look at it as an opportunity to improve, an opportunity to find something new. All right, let's go on. Let's go to the next experiment. Let me pause for a second. All right, so now we're going to go ahead and move on to Meselson and Stahl. Now, Meselson and Stahl, they knew DNA was the inheritable material, right? So at this point, we all knew that DNA was the stuff, it was the coding of life, and we knew that DNA had to be replicated. So they knew that they weren't sure how the system worked yet, right? But they knew that there's this thing called the DNA molecule. They knew there was this thing called a chromosome, right? And they knew that it had to be copied. They knew they started with one, and they ended up with these two copies connected by what? Centromere. That's right, a centrosome. These two copies connected by a protein called a centrosome. Yes, they knew there were histones in there that were wrapped around somehow, but they weren't sure how because they didn't get a good picture of it. So here they are. They knew this was going on. They knew there was a centrosome. They knew there was these two copies connected, and they would eventually get split off into a new cell. We'll cover that next week when we do mitosis and meiosis. So that being said, they questioned how did this happen? Like, did this? So there were three options. They thought there were three options to this. And now this is going to be important that you understand what we're talking about at this stage. If you don't understand this stage that I'm about to go through, you're not going to understand the rest of the experiment. And I warn you, after we went through it, I think we spent an hour last night, a bunch of us after school, during our tutoring session, going through this. And at every stage we said, okay, you need to know this. If you don't know this, you're not going to understand the next step. And that's true of this whole experiment, which is why some of you are confused, right? And why we're spending this extra time going over it. So, all right, so there's a couple ways this could happen. I'm going to do two. I'm going to leave the original strands in pink, all right? And I'm going to change the new strands.
What's the circle in the middle?
After school.
No, we went over this yesterday in class.
Yeah, we did, but I want to break it down. Is that okay? Because there's still people confused. I wanted to break it down. We did go over it in class. Yeah, you're right. You're right. We did go through it. But I just want to make sure we're all clear on it. Because I think there's some confusion that might lead to wrong answers on tests. Is that okay? Yeah. All right. So I'm going to put the original strands now in pink. And what that means is that when I say original is that we're going to copy these, right? So somehow the question is somehow these two strands were copied. The question is, are these two strands here in one molecule? Are they separated or are they somehow dispersed? So now let's take a look at this. So one way that this could happen, one way that this replication, we're going to call it replication, right? It's a study of DNA replication. This word replication means what? Multiply or copy. Or copy. To replicate something is to copy something. So one way that could happen is that you can, I'm going to do kind of half steps. You can open up the DNA molecule, right? And you can start to copy a little at a time. So start to copy here, and you start to copy here, okay? And then what happens is that in the end, you end up with one old strand, right? You see how that's happening? You see this? And we're going to go into detail on it next week. You see here you have a new copy in white, a new copy in white. And this is going to keep moving in this direction. Let me use a different color. In this direction here, and the other one's going to move in the opposite direction. So in the end, you're going to end up with, right, that's right, it's semi-conservative. So you end up with with 2 strands, right? Can you imagine that? Can you imagine that you open this up and you start to copy each one, and so that in the end, you end up with kind of these 2 strands, these 2 new molecules, or whatever you want to call them, these chromosomes, right? All connected by this centrosome, and we call this semi-conservative.
Well, I got a question.
Yeah.
What is centromere? Because I began confused with all this thing.
Right, I agree, and I warned you, right, that was going to be a confusing point. The centromere is this thing here that's connecting the 2 chromosomes.
But you said centrosome.
Did I say centrosome? I'm sorry, that was my mistake. I meant centromere. Centrosome is something new, different. We'll talk about it in a minute, and when we do mitosis.
So that's centromere.
Centromere, that's right.
That connects.
Let's just talk about centromere. Let's not go anywhere further. Let's just do that. So this is centromere. Everybody okay with that? Connecting 2 chromosomes. Thank you for correcting me. Now, so that's semi-conservative. Everybody okay with that? All right. So that's one step, and that's important that you know it. And by the way, this is how it works. Okay. These scientists, Meselson and Stahl, did not know that. Yeah.
It's not. Oh.
How all life does.
Here you go. It says how all life does replication.
Semi-conservative is how all life does replication, all right? So let's be clear on that when we're talking about, interesting. When we're talking about this version right here of how it does it, this is how it does it. Are we clear?
Yeah.
All right. But Meselson and Stahl did not know that. And so they thought this was one way. Then there was another way they thought it could happen. You could have your original strands, right? It's your original DNA. Instead of old, I'm going to use original, right? Because we found out last night that old got confusing as we went through the steps. So another way we could do it is we could copy it and end up with the original strands still together, and then the new strands together. Now I want to point out something. Do you see how there's 2 new strands in white and 2 old strands in pink in the semi-conservative model, right? But notice that there's one original and 1 new for each chromosome. Where here, for each chromosome still connected by a centromere, each chromosome has the original strands together and the new strands together. So because we are conserving the original strands, we call this the conservative model. Huh?
So how is that for all life?
This does not, it's a good question. I'm glad we're doing this, because that's exactly the problem. People think that these models all exist. They don't. This was a possibility in the beginning. We know now today that this is not true. What we're going to go over today is the experiment that proved that this is, or that showed us that this is not true. So this was one. Let me write that down. Let me use a yellow and say these were possibilities. All right, in the past. This is no longer true. This is no longer true. Conservative model, and I'm going to go ahead and put a little asterisk by it and say, not true. All right. It's not the way it's done. So they said, okay, there's another possibility. So here we have the conservative model, right? And we have the, what's that? We have the conservative model and we have the semi-conservative model, right? But then we had another possibility, and that's you had the original DNA, right? And we go through replication, but now instead of either the chromosomes being all the original strands and another one being the new strands, or one chromosome having half of each, one strand old, one strand original, one strand new in each molecule of DNA. What the other possibility called the dispersion model was that you would have a partial. You'd have partial. You'd have partial. Hold on a second. For each one, you'd have all right, so you'd have this centromere connecting two DNA molecules that were hybrids, almost Frankensteinish, right? Where you have like pieces of the original and the new DNA in each strand. All right, so we call this the dispersive model. Only one of these models would be correct. At the end of the experiment, only one of these models would be correct. And we know today that that dispersive model is not true, not correct, right? But we're going to look at the experiment. We're going to look at the experiment that showed that the most likely truth is that semi-conservative is the way that we do DNA replication. Today it's clearly semi-conservative. We actually use that understanding to manipulate DNA, so we're pretty sure. I mean, we're almost absolutely sure that this is the way it is. Yeah.
What did Joe Meselson and Stahl even make those possibilities? Like, would that those other models today?
Honestly, that's probably one of the most important questions that this whole experiment, right? Like, what made him think that these were even what made him think that these were the possibilities? All the experiments that came before theirs. So all the knowledge that came, all the different labs, they go, we get to, as scientists get together in conferences and they meet with each other, they make presentations. You do presentations for your experiments and science fair and that kind of stuff. Well, scientists get together in conferences and they present their research to each other. They discuss it. They call each other. Someone in Japan calls you in England and says, hey, I'm doing this experiment. This is what I'm getting. What are you getting? Because I know you're working on something similar. And even though that does happen, there's also competition. So there's a little bit of both things going on across labs because there's a competition for resources, for money, to do research. And so all this is going on in this environment, but every conference they get together and we share. And then finally somebody publishes something. So write this down, 2 major journals. There's a lot of journals. Every subject has their own journal. But there's a big journal called Science, another big journal called Nature. Those are kind of the 2 biggest journals in the world. And journals are like magazines, okay? But what they do is they publish research from scientists. And before they get to publish the research, scientists get to review it. So I think enough that we go over it. So you have a journal, you did some research, right? Just like Meselson and Stahl did. and you're ready to publish it in the magazine. You send it to the journal. The journal sends it out to 3 different people. 3 big, sometimes more, but 3 big named people to review. They're going to review your work. They're going to pick it apart. Who watches Ink Master or Chopped, right? Skinned, right? And so what they do? What do? Somebody makes something, right? And then there's a panel of people that review it, right? And they critique it. And that's what these guys do. They critique it. And if it's good enough, they say, okay, go ahead. If they get the go ahead from these we call these peer reviewers, right? You can think of the peer reviewers as almost judges, right? Like an Ink Master. And the journal then decides, okay, print it, and they put it in their journal, and they publish it, and other scientists get to read it. So based on that, Meselson and Stahl said, after all their readings and all those different journals, they said, okay, these are the 3 possibilities, let's work on this. So it's a really great point. It's really one of the more important, because in science, nobody does anything on their own. Everything you know from people that came before you, from your academic fathers, from your academic grandfathers, from your alma maters, from your journal reading. We learn from each other. Hundreds and hundreds of years of learning. That's the power of science. And remember, in science we don't say we understand it. We just say this is what we think is going on right now. We'll see what happens tomorrow. Okay? All right. So that's where Meselson and Stahl we're looking at. Those were the 3 models. Everybody got it and understand it, right? Because I think there was some confusion on that. All right. Now, here's some facts that you need to know to understand this idea of a petri dish. Everybody know what a petri dish is, right? Who doesn't know what a petri dish is? Why does this thing always do the biggest one? I don't understand. All right, so a petri dish is a plastic dish, and I think you know it, you just don't remember it. It's a plastic dish where we grow bacteria. You remember that? You never seen that? Are you serious? You never? All right. Okay, it's not about school districts. You've seen this on TV, I'm sure. But okay, you know what? Okay, I'm going to show it to you. Everybody, chill out. Here is a petri dish. So you see a petri dish looks like this. You've seen this, right? Oh yeah. Oh that. Do you see? This is actually pretty cool. You see the bacteria growing? Do you see it? These are bacteria. You see them growing? Okay, so we'll talk about it later. We can do it in class if you like, but we have to be very careful. All right, so we're not going to get lost in this. So what you understand is that each of these is what? What are these things growing on here? Bacterial colonies. There's millions and billions of bacteria in there. All right. So it's not. Well, let's say millions of bacteria in these colonies, depending on how big the colony is. Everybody agree? Understood? Bacteria are not that giant, right? They're very tiny. So from this petri dish, there's a couple things you should know about the petri dish. Where did the petri dish come from? Yeah.
Prokaryotes.
The petri dish? You're right that the prokaryotes are growing on the petri dish. You're right. I love it how you guys keep answering questions you're not asked. You get so excited. I forget how excited youth get. You guys remind me all the time. All right, so let's start again. Where did the petri dish and the food? You know we're growing it on food. You know the petri dish we fill it with solid food called agar. It's called agar seaweed, and then we put food in there like protein. Sometimes we can even grow it in blood. We can put blood in there. So it's really pretty interesting. So there's food in there, right?
Yes.
Where'd that food come from? You put it in. You're making the petri. You as a scientist are making the petri dish. So you make it and you sterilize it, and then you take bacteria, right? In this case, it doesn't matter what the bacteria is, but you're going to take the prokaryote, as someone said loudly. Yeah, the Petri dish is the plastic thing, you're right, but inside the plastic thing we put food. Yeah, huh? I can't hear her question, please.
You know how in the picture it shows like little spots?
Yeah, the spots are the bacteria. Right, but the spots are growing on something. They have to eat something. And we put that stuff in. It's called agar. All right, so what it is we take the Petri dish. Can I turn that inside? Can I turn that like something like this? Is that all right? And then I'm going to fill it up. I'm going to take a beaker. That's a horrible beaker, so I'm going to redraw it. I'm going to take it. It's probably not going to be much better. All right, so I'm going to take a beaker, and from the beaker, I'm going to fill it up with first a liquid, like Jello, almost like Jello gelatin. I'm going to fill it in with something called agar. There's several mediums. You don't have to use agar. That you can put blood in it. You can mix the agar with blood. You can mix it with proteins. You can mix it with antibiotics. You can mix it with whatever you want. You're making it, so put whatever you want in it. And it's like Jello gelatin. You can have strawberry Jello gelatin. You can have cherry Jello gelatin, right? So we're going to add the flavoring. Instead of flavoring, it's actually going to be food. You see, and then on that agar plate, you're going to place bacteria. You're actually going to start growing bacteria. They're going to grow. See the little bacteria growing here? These are colonies.
Are you creating an environment?
You're creating, it's perfect, yes. You're creating an environment for the bacteria to grow. And what's cool is that you control what you put in there, right? No. I do somewhere, but I'd have to look for it. So, yeah?
So if it's making its own environment, then don't it control whatever scientists put in, don't it control what it'll make bacteria?
Exactly. That's the point. I'm glad you got that. That's perfect. That's right. That's the key. She just said the key. Because you're making the environment, you control what the bacteria have, right? And that's the important point I'm trying to make here. The scientists had control. So,
But does the scientists have control now? Since you said that they can help
I'm talking about in the experiment. We don't control the world. That's nature. But in this experiment, scientists control what's going in here, right? That's the food. That's what the bacteria have only what's in that agar to grow. If you don't put it in there, they don't have it. If you put it in there, they have it. Is that clear? Because that's important for you to understand the next step. Yeah?
So, you say you can put blood in there for that. Is that what happened when bacteria is in their cells and stuff?
Yeah, well, the reason we would put blood in there is blood is a very good, some bacteria grow very well in blood, and we want to see how well some certain bacteria grow in blood and how they don't grow in blood. So yes, it's to study the impact, the growth rate of some bacteria in blood. Some bacteria need blood to grow. So it's interesting. And parasites, some parasites. And it doesn't have to be, by the way, this food is solid. Agar turns into solid like, or kind of it's actually a colloid. You should know that from our previous studies. And a colloid is like Jello gelatin, right? So it's a gelatin. It's kind of a solid / colloid, and it's a Jello gelatin that the bacteria grow on. It's made of seaweed protein. It's irrelevant. Can we move on? Because it's not really the point I want to hit on the experiment. Yeah. That's exactly right. But we're not concerned about that in this experiment. You know what? That's another essential question, Andreas. It's like you have to think that all the other experiments that we may discuss in the year, that's going to be key. That because you can control what goes in there, you can say, I'm going to grow this kind of bacteria, I'm not going to grow this kind of bacteria, and I'm going to see which bacteria grow better on this and which bacteria need what. Okay, so yes, you can use this fact, right, that scientists, that you control what goes in there to study all kinds of things. But now, in this particular experiment, we're looking at DNA, right? Right? Right, and one of the things that DNA definitely has in it, without a question, that everybody should know at this point, but I'm going to go ahead and draw it because it's going to be on the test, and I expect you guys know it, so I'm going to go ahead and draw it again, is that I expect that you understand that DNA is made of a sugar, a ribose sugar, there's an oxygen here, that's right, very good, and a phosphate, right? So the phosphate is here, and I expect that you understand that DNA, and this goes for RNA too, but DNA also has this nitrogenous base coming off here. I don't know, I just say, you got this nitrogenous NB, nitrogenous base. In other words, what's in this base? Nitrogen. So, as long as you understand that you have this, that there's this nitrogen sitting in here, in every one of these DNA molecules, you have nitrogen heavily in there. So you control what kind of nitrogen is in that petri dish, don't you? Yeah. So, that being said, the scientists want to find out what? What's the purpose of the experiment? What is it? What does the scientists want to find out?
How DNA is structured.
Close. How DNA replicates, right? So, all right, there's one more fact you should know, and that's that these bacteria are going to replicate, they're going to copy. They're going to replicate every 20 minutes. So you got 20 minutes, and you're going to get a new set of DNA. Where's it going to get the nitrogen to make a new DNA? The food. So it's going to get the nitrogen from the petri dish, right? From the agar. The nitrogen that you put in the agar, right? All right, so that's going to be very important, all right? So let's take a look. Now we're going to have to ask if-then questions. Let's look at if the semi-conservative model is correct. So if it's correct, let's look at that. If the semi-conservative model is correct, then what's going to happen? Then the following should happen, right? So pay attention, let's look at this. Hold on a second. A couple more things you should know. And we have two forms of nitrogen. We have nitrogen 15, which is heavier or more dense, right, when it's in a molecule. And we have nitrogen 14, okay? It's going to be lighter. We can put it in a test tube and spin it in a centrifuge. The centrifuge spins around, okay? And this test tube is going to be filled with different liquids of varying densities. Now, what do less dense things do? Flow. So that's important. So what we can do is we can say, okay, the real dense stuff is going to be in the green layer, and the less dense stuff is going to be in the yellow layer, and the less dense stuff in that is going to be in the blue layer, and then we can say that this stuff even less dense than that is going to be in the pink layer, okay? And is class almost over? All right, guys. So we'll finish this tomorrow, okay? We'll move the test to next week. Look, if we don't understand this, if you don't understand this, it doesn't do any good to give you a test, right? So I just want you to understand this before I give you a test. All right. You're welcome. Right. So the semi-conservative model is correct. This is the next day. And here's your density column. So you can create a density column in a test tube, right? Each one is a different density, the least dense and the most dense, right? Then when you add, if this is, I'm going to give them numbers. I'm going to give the least dense numbers. I'm going to say the least dense is, let's say, the same units. I'm going to use the same units here, the AMUs. So I'm going to say this is, something like, let's say 13 AMUs at the top. Then this is 14. Then this is 14.5. And this is 15, and then this is 16, okay? So those are your layers. Everybody get that? All right, so I'm going to put, remembering that, because we're going to use this, and by the way, what we do is we just drop the DNA in there, the cell structure in there, and then we have this thing called a stain, or another word for it is a dye, and it's called a DNA stain. DNA stain. So it's going to stain the DNA black. You can't see DNA, right? I mean, if we isolate DNA, it might look a little cloudy if you have a lot of it, but it won't be any color. But we can add a stain that only binds to DNA. Is that everybody okay with that? So wherever there's DNA, boom, there it is, right? So knowing all that information, I don't even know what that said, but I don't want to know. All right, so if the semi-conservative model is correct, right? If it's correct, then we should be able to predict some things, right? So here's our predictions. There's that idea of prediction again. What is our prediction of what's going to happen, right? When we take this Petri dish that has these colonies of bacteria, right? And remember, the Petri dish has all the food source. And we grew it with nitrogen 15. This is what we grew it with, normally. So all the DNA in there has what? What does all the DNA in there have? Someone said it, nitrogen 15. Okay, so if we have any DNA molecule we take out of there, is going to be based, and remember from the previous slide that it has all the DNA has nitrogen in it, it's going to have nitrogen 15. So if we threw it in the density column, right, it would show up at this point here, wouldn't it? Does that make sense, everybody? Okay. So now another thing you have to know is that every 20 minutes, you're going to replicate this DNA. You're going to make a copy of it. So after 20 minutes passes by, out comes 2 copies of the DNA molecule, right? So that new copy, if we move it to a new Petri dish, if we move these colonies to a new Petri dish, what do you think is going to be on that? What are we going to put in the new Petri dish? Say that? And 14, that's right. We'll put... Did it? Look, can I have the experiment, please? Let me have the paper. I might be wrong. Can I see that? There's one right in front of you, darling. So it might have been nitrogen 14 first. That makes more sense, maybe. Well, I don't know. We have to look at the experiment. What did the experiment show? They have controls. We have controls 9 to 14 and 15. But according to this experiment that we have, since bacteria in the laboratory can grow on plates of both cultures exposed precisely defined mixtures of chemicals, Meselson and Stahl grew bacteria in cultures containing only nitrogen 15 for many generations, so that the DNA was almost entirely composed of nitrogen 15. These chromosomes are now more dense than nitrogen 14 chromosomes, and spinning chromosomes are now very high speeds, heavy chromosomes further down, blah, blah, blah. After growing the bacteria nitrogen 15, producing bacteria with heavy chromosomes, they shifted the bacterial growth to conditions in nitrogen 14. So 15 was first. Is that okay? Yeah. Yes, go ahead. So we started with nitrogen 15, and now we moved the bacterial colonies in nitrogen 14. Let me pause for a second. That's right. Nitrogen 14 was you moved it to a plate with nitrogen 14. You wait 20 minutes, right? What's going to happen? The new strands, that's one round of replication. That's right. So what we expect to see, right? If it's again, this is we're testing the model, right? If this model is correct, and we take them and grow, and we have nitrogen 15 bacteria, with nitrogen 15 size density chromosomes. And we grow one round of replication, let them do replication once with nitrogen 14. Then what would we expect to see? If it's semi-conservative, one of each of these molecules will have one, let me make it a little wider. Each of these molecules is going to have one of their strands be nitrogen 14. Is that right? Why is it nitrogen 14? Why is this new strand, we're going to call these the new strands, why are the new strands nitrogen 14 where the original strands were nitrogen 15? Why do we say that half the DNA is going to be nitrogen 14? Because we grew it on the nitrogen 14 plate. Does that make sense? Because that's the only nitrogen they had to make the new DNA. They were making new DNA, the bacteria need nitrogen, they got to take it in from their food. Look, you're a growing person, you're a baby, you're growing up. What kind of nitrogen do you have to build with? Whatever nitrogen your mom feeds you, right? Whatever food your mother feeds you, that's what you have to grow with. What you eat is what you have to make yourself. Does that make sense? So the new bacteria had to use the nitrogen 14 because that's the only food source they had. So the nitrogen 14 was incorporated into the DNA. So now you have what? Where this one was not. So let's go ahead and flip to the next. It's going to get kind of crowded. So what we had is originally we had this DNA molecule with all nitrogen what? Nitrogen 15. And then we went through 20 minutes of replication, so one round of replication. We still had, this is semi-conservative, right? This is a prediction. This is what we would predict would happen. If this is correct, then what we would expect is that the new strand, because we moved it, remember we moved the DNA, the colonies to the new nitrogen source. The new strands would be half and half, right? Nitrogen 14 and half nitrogen 15. And let's say again, nitrogen 14 and half nitrogen 15. Everybody okay with that? Does anybody have any questions, concerns? Yeah.
You know how you said there's one strand of N14? I'm confused because if they had one row of DNA like on the N15, what if their order was like C, G, A, F? What would the other side, wouldn't it have to be the opposite? I'm confused. How would it happen?
Okay, so then to answer that question, what you have to do is you have to remember what the DNA is made of. And that goes back to knowing your material, right? You have to remember what DNA is made of. DNA is made of, and you're going to be tested on this, so you need to know. Your ribose sugar, your phosphate, right? And what? Nitrogenous base. This is your nitrogenous base here. But the nitrogenous base, C, G, A, T, all of them have what? They all have nitrogen in them. I'm not going to draw the bases out. But just know that they have nitrogen in them, okay? That's where your nitrogen is. So doesn't the order of them, which is I think what Burson was saying, the fact that this is A to T, G to C, right? The order doesn't matter because we don't care what the order is at this point. We're not trying to find the gene. We're not trying to find the code, right? What we're asking is how do you make the copy, right? So we're going to get into the copy DNA replication next week. But what you need to worry about right now is that the question is, how do we copy it? How is it that we copy it? And remember, the semi-conservative model predicts that what's going to happen is that this thing, and if we're going to break it up into steps, that this thing is going to kind of unwind, all right? And as it unwinds, it's going to make a copy in this direction and a copy in this direction, so that you get one copy, you get two copies, one with an original strand, right, and one with a new strand. Is that clear? And we said, this is the way it's done. This is actually the way it's done, but I want you to understand the experiment. I want you to do your best to understand the experiment. The test will have this experiment, all the steps tested. I will be testing this heavily. So you have to understand what this means. So you have this new nitrogen 14, nitrogen 15. So what does that, if this is true, knowing what we know about density columns, and the fact that we can stain them, right? If nitrogen 14 would be found here, right, and nitrogen 15 would be found here, all right, where would we find these new chromosomes? In the middle, that's right. So what we would find is that if we're, I can't use black, so I'll use white. If we were trying to find the band, remember we'd stain it, right, so that we can see it, then we would find the new strands after one round of replication, we would find it here. This is if it's semi-conservative, right? If it's semi-conservative. All right, you'd find it in the middle. The original one, this original, if we took the original DNA and ran it through a density column centrifugation, we'd find it down low, wouldn't we? So we do control groups. So we have to do a control group. Our two control groups are what would it look like if it was just nitrogen 14, the band would be up here, right? And if it was just nitrogen 15, then the band would be down here, right? But the new strands, after one round of replication, guess what happens? It's in the middle, so that kind of supports semi-conservative. But what would be the prediction of the other two models?
It would be the old mast cells and the
That's right, so let's take a look at it. So let's take a look at conservative. For the conservative model, what we would expect is you'd have a DNA molecule that looks like this, and after one round of replication, we'd have the original strands together. That's what we mean by conservative. The original strands stick together. And we'd have a new strand, trying to keep the colors the same, we'd have a new strand that looks like this. A new DNA, a new chromosome, right? And yes, they're sister chromatids, but after they get separated, they would be chromosomes. So this new strand would be nitrogen 15. The whole thing would be nitrogen 15.
Wait, you mean the new strand would be 14?
Oh yeah, sorry, my bad. I keep messing that up. So the new chromosome would have two new strands, right, of nitrogen 14. The original chromosome would have two strands of nitrogen 15, right? And so what would you expect to see inside the density column test tube?
A band.
Two separate bands, that's right, good. You'd expect to see one up at the top, and you'd expect to see one down at below, and nothing in the middle. But we don't see that. That's not what we see. We see, after one round of replication, what we do see is a band in the middle. So because we see that, this is what?
Wrong.
Disproved. It's wrong. Because remember, in science we disprove. So this is disproved. That this is not the conservative model is wrong. Yes, ma'am.
If science is disproved, then how, when something works, like, is that like, you know how some people find out like like what made everything come together, but they use what they started from other people's experiments and stuff. So how does that work? How do everybody get recognition if the one person who's made the breakthrough? Like how did
Everything, it's kind of complicated, but I'll say this, that recognition was given to Meselson and Stahl. They got a Nobel Prize because they made the breakthrough. The people that supported them, the people came before them, they got paid salaries, they got published in magazines. When they go to conventions, I'm sure Meselson and Stahl were like, man, we used your research, thank you very much. But there's always only one winner, right? When you make an album as an artist, you have sound people, right? You have support, you have assistants, you have a whole entourage, right? But only the artist, the only person you ever hear about is Beyonce. You never hear about the 100 people that help make her who she is. You know, the person who actually tweets, because you know Beyonce's not on that phone tweeting, right? You know that she has people that tweet for her. So there's all kinds of support that doesn't get necessarily credit, but they're part of the team. But the people with the ideas, the people that make the breakthrough, they do get the credit. And the reason we as teachers teach you these people is because, yes, they were very smart, they were very innovative, they came up with the invention, but what's really important is what? The idea, right? The process. So really that's what the important part here is. Yeah?
Okay, so when we talk about dispersion, would there be like multiple bands?
That's a good question. Let's look at it and see what we would expect. You tell me after we walk through. So let's take a look at conservative. So conservative is out, right? Semi-conservative matches the results. The prediction matches the results. So we're leaning in that direction. So now let's take a look at this dispersive model. Now in the dispersive model, as you know, you start off again with a molecule of what? Nitrogen 15. And again, you're going to move the bacteria to a plate that only has nitrogen 14. You're going to wait 20 minutes. But what the prediction, again, I want to emphasize that this is a prediction, right? The model predicts something, and the model's predicting that what you're going to get is two molecules that are evenly made, randomly made of pieces of DNA of both nitrogen 14 and nitrogen 15. So there is the replication is going to be, so you're going to get two of these, and I like to call them Frankenstein chromosomes, right? Because they look like pieces and parts. But it's going to be about 50-50 of the DNA in both of them. All right, so you got these two molecules that are each of the strands of the DNA. Each of these strands are made about 50% nitrogen 14 and 50% nitrogen 15. That's each of these strands, all four of them. All right, 50-50. So what would you expect if we take and we put this in a density column? What would you expect? Would you expect to see two bands? Where would they be? Would they be in nitrogen 14 or nitrogen 15? It'd be in the middle. As someone said, it would be in the middle again. That's right. It would be right in the middle. So how does that tell? How do we tell? Since the band is actually in the middle, right? After we stayed in, that's the result. When we test it, it actually is in the middle. Is it dispersive or is it semi-conservative? Dispersive. Well, wait a minute. Yeah, it's the same thing. The dispersive and the semi-conservative predict that you'll have a band in the middle, and the results from the experiment is you have a band in the middle. So after one round of replication, you can't tell the difference. So we got rid of conservative, but how do we know it's semi-conservative and not dispersive? Well, this is the trick. We wait one more round. We wait another 20 minutes. So now, I'm going to try to put both of these at the same, semi-conservative, and I'm going to put dispersive so we can compare the two. So semi-conservative and dispersive, let's take a look at what it would be predicted in the second round of replication. So we're starting off with not one nitrogen 15, right? What are we starting off with in the semi-conservative? Right, we're starting off with that one nitrogen 15, one nitrogen 14. And the dispersive, we're starting off with that kind of, if you don't mind me saying, Frankenstein-looking chromosome, where we have a little bit of each of the two nitrogen sources. Agreed? All right, so what would happen in the second round? Second round of replication. So now 20 minutes have passed. Now you're going to wait another 20 minutes. So you see what happens now. What's going to happen after replication here?
It's gonna be two of the same thing.
Okay, so they're going to open up, right? And you only have nitrogen what? You only have nitrogen 15. No, I'm sorry, 14. Only have 14. You only have the red. You only have the red nitrogen. So you still have the original. You still have this original. So you only have red. So what's going to be the other new strand? What's going to be the new strand? Red. Because that's all you got. You only have nitrogen 14. But the other strand, remember this is the second round, right? What color is this? It's red. It's nitrogen 14. That's all you got. So you're starting off, because that's what you had from the first round. This is what you made from the first round.
Right, but I thought it was a 15 and a 14 for semi.
Right, good. You had a 15. There's your 15. Right? Because each one of these is going to be a master, and the copy is going to be made from them. Okay? But what about the other one? This one makes sense to you, right? Because it started with this one. But this one now has to be copied. But it's going to be copied. What's the only color? So it's going to still be there. It's not destroyed, right? So it's red. And what's the copy? Red. And so what you're going to... I don't understand why you think... I understand where you're going wrong. I'm trying to figure out where exactly your confusion is.
Because this is second one, right? So we first start off with 15.
We start off with this one.
14.
They're starting right here. This is what we're starting off with. Because we ended the first round with this, right? Which is 114 and 115 and one chromosome. So what we ended the first round with, we start in the second round.
I'm confused on why the two 14s now.
Well, I'm asking you to think about it. You started, huh? You want to try to answer that? Yeah, go ahead.
Because when the first one was made, you used the last 15, and all you had left was 14.
Well, I wouldn't say it that way.
How could you make 3 copies of the 14 then?
Because they were mixing it.
You're making nitrogen, you're making it. You're making the copy. You're reading one and making the other.
15 is just 15 is not,
It's from the original.
right, so it's not there anymore.
No, it's always there. It doesn't go away. It just gets copied.
So why that one is all red then?
Because they were mixing with
All right, well, I'm going to try to start up again in a minute and see if maybe I skipped a step and I didn't realize it. Just hold on. Yeah.
Okay, so DNA is semi conservative, and like you have to go. What about some people who they say like has like genes from distant ancestors and stuff like that? So how does that happen, or does it just stay recessive during that time and it only comes out from them?
Okay, so that you're skipping like 3 or 4 steps in that question. You can't understand that until we understand what DNA is. All right, so we can't jump to that. So let's start here as a class. Let's go back. Raven's asking a question I bet you is in a lot of people's minds, and you're just not voicing it. So now could we try to figure this out together? All right, so I'm going to go ahead and shrink this up a little bit. And I'm going to say, look, guys, when you do DNA replication, you have these 2 original strands, all right? They're both native nitrogen 15 in this experiment because that's what they had. You open this up. You open up this strand, so it goes like this. And it's called the replication bubble. You'll learn about it tomorrow, right, next week. So you're going to make a new copy, a new strand. This is the new strand. These 2 go together. That's 1 chromosome. And then these 2 go together. That's 1 chromosome, right? So you notice they were half and half. That's what you get. That's what you're starting off with here. You see it? Now, if you already have one, look at what happens. Pay attention. That's the first round. In the second round of replication, you start off with this. You're starting off with this. Does everybody agree with that? Okay. So now we're going to go ahead and make a copy of this, That means that this is going to open up a replication bubble, but what do you got? One strand is nitrogen 15, right? And the other strand nitrogen 14. What is it that what nitrogen do you have in this experiment to make the new strand? And 14. Only red. You only got red to make the new one. These are your two old ones now, right? So now let's make the new strands. You make one like so, right? That's one chromosome. And what is the other one? Do you see? That's why. Because every time in the semi-conservative model, every time you make a copy, you're using one of the two strands as a template, and you're using you're making the second one.
Wait, so how would the next one go?
Well, okay, so that's the next. So in the third round, if you're going to do a third round, and it would keep going, okay, what you're going to have is you're going to start off, unfortunately, you're going to have to say, you're going to start off with two types, right? You're going to start off with this, and you're going to start off with what? This. And so when you make copies of those two, you'll get one that's, again, you'll end up with 4, right? One like this, and 3 that are what? All nitrogen 14. The bubble thing, it's here. It's the same thing that's happening here. So you just take these, open them up, and then you're making copies. Is that clear? All right. So, I don't know why this thing makes noise sometimes, not all the time. Is someone touching them with their feet? Push this up. Oh, yeah, there's some kind of tension with the wires.
That's positive feedback.
No, I was just about to say that.
Yeah, just try to stay away from those wires, please. All right, so I'm glad that we're getting it. I hope you get it. Pay attention, because you're going to get these questions, and it's going to really help you a lot when we do mitosis and meiosis, okay? Pay attention, though, we're not done. So what's predicted from the second round of replication is that you'll get one molecule, one chromosome with nitrogen 14 and nitrogen 15, and one chromosome molecule, one DNA molecule or chromosome with 2 nitrogen 14 strands, right? 2 nitrogen 14 molecules. That's the prediction. And if we're going to draw out the prediction if this is the predicted, how many bands would we say would appear in the density column?
2.
According to the semi-conservative, we'd expect to see, that's right, we'd expect the 14, which is up at the top, and 14 and a half, something in the middle, and no band at the bottom. That's what we would predict. Okay, good. Now, if we did the dispersive, hold on a second. Okay, so what we have here is, if we have dispersive, then what we'd expect to see is what? 2 bands, right? 2 strands, rather. And each strand having a combination of 14 and 15, right? So that's kind of what we'd expect to see. In something like that, right? We have both. So how many bands would we expect to see? The predicted in the density column, we'd expect to see how many bands?
3.
Where everything's 50-50 here. Why would we expect to see 3?
It's like 14 top, 15 bottom, 1.
Okay, yeah, you're right. If we had an original 15 chromosome, then we had a 14, then we had a mixed one. But what are these 2 chromosomes? Are they all 14, all 15? No, they're in the middle, right? So we'd expect to see what? 1 band in the middle. You see, in this model here, we predicted 2 bands, one at the top and one in the middle, because one chromosome was made of the light nitrogen, right? And the other chromosome was made of half heavy. Where these 2 chromosomes in this model, it predicts that we would have one in the middle, because both chromosomes are made of 50-50, heavy and light. Yeah,
So the dispersion method was only right once.
that's right. Good. You skipped a step, but I'm good. Let me catch everybody else up. But he's right. What Steve said, Steve knows what the results were. And the results of the experiment, the actual results, these are predicted, right? If dispersive is wrong, then there'll be more than one band. If semi-conservative is wrong, then there would be only one band. But what we see in the actual results is actually one at the top and one in the middle. So this means that the dispersive is disproved. So the dispersive is disproved, it's wrong. So that leaves us with the semi-conservative one. This is the one we accepted. We go ahead and accept the semi-conservative because all the evidence, all the experiments point to that model being correct. All right? So this one is wrong here. So I've said it like a million times, I hope it sinks in. Dispersive is wrong, semi-conservative is right,
Conservative.
and conservative is wrong because the evidence clearly shows that the model cannot predict the actual results. Yeah.
Who would the replication bubble for the second round of the dispersion method?
It would be just way confusing. I'd rather not do it if you don't mind. We can do it after school if you'd like. When we go into DNA replication, you're going to see the replication bubble. We'll see all the enzymes and everything. But right now, I think I have the class pretty much understanding this concept, and I'd like to keep it that way and keep it at this level for the exam.